Right. So for research design, I mean, it is the same thing. Research design, experimental design, for me, is the same thing. Okay. So this is when we talk about problem statement. Yes, it's already a hard work, and then comes in literature. It's two also. It's actually very time consuming. I hope that you have feel you now that you feel the the burden of reading and trying to sort your readings and trying to filter all the informations that suits you, okay? And you have gone through the training offered by the library, okay? So these are very difficult steps to do, eh? But as I said at the beginning of our discussion, that it depends on whether you have a very large scale research project or you're talking about small scale research project. So the work is relative. So if it is a small scale, then the literature is smaller in scale, and the problem refining also is smaller in scale, as well as the research design. Okay, so it is relative to your length of study, scope of study. So when we talk about research design, we're talking about what we want to do. What we want to do. We have an idea to solve, and we feel confidence that the solution that you wish to propose will work. I mean, you have that confidence level, high confidence level on that your ideas. But what to do now? What do you want to? How do you want to start? Okay, right. So you can see that these people are talking about talking about the study itself. Okay, so you can see the word expensive, and you see the word takes too long. So you can guess that whenever that we are designing experiment or research, okay, there are a few factors. One is, of course, money, the financial part, and the other one is time. Okay, of course, on the novelty, on the significance of the problem, we have settled that. We confirm that we have the novelty, as well as we have the problem is real, is very significant. That one we have settled. But the solution, we have the solution in general, and how do we design the experiment so that we can answer the objective. Okay, so we design experiment to answer the objective. Okay, so that's why it is related, whether you want to have only one objective, whether you want to have 6 objectives, that is related as well with your experimental design. So now that you decide your master dissertation will be 1 year, okay, and you have really you have actually decided on the scope of your study. It's going to be a very small problem, okay? So you also have to look at your how much money do you have and how much time do you have. That will be enough for you, okay? That will be enough for you. But for those, for researchers like me, okay, we have to talk about monitoring, okay? How am I going to be monitored based on that particular proposal? If I propose and the proposal is granted, is accepted by the stakeholders, for example, and how will I be monitored? Is it going to be based on the 3 months monitoring, or is it going to be based on 6-month monitoring? Okay, so that one I really have to consider, because let's say if time comes, it's already 6 months, and let's say 3 months or 6 months, and I do not have the data to present to the stakeholders, then I'll be in big trouble. So for you, you only have to consider Whether when you talk to your supervisor say, yes, I do have enough money to do this. And then the time, you have enough time to finish the experiment as well as the reports. Please remember that you have to write reports, then submit on time. Okay, that's why for me, okay, when we talk about experimental design or research design, and you have decided this is your life, this is your life going to be for another 3 semesters, this is actually your life design. Okay, this is actually your life design. How do you design your life? Because it will take a lot of times, and actually this is really related to your life. Okay, so I always talk to the students, this is not about experimental design, it's your life design. How do you design your life within this 1 or 2 years? Okay, because you're going to be in it, you're going to be it's going to take tolls in your life. Okay, so it will take a lot of your time. Okay, so when we talk about your life, of course, you have to really sit down. Yeah? You have to really sit down and think. Okay, you cannot just write it on papers. Okay, and expect everything will be smooth. Okay, all right, the first month I will do this, the second month I will do this, and the third month I will do this, fourth month, and everything completed. Yes, I will be happy, and now I can do something else. Yeah? There are a few factors that you have to think about. Let's say you want to do preparation of nanomaterial, for example. Yeah, you want to do preparation of nanomaterial, or let's say you want to do something related to natural product. Yeah, so natural product requires you to do extraction, a lot of extraction from natural resources. And then you need to submit to the samples to do GCMS, NMR, or any other characterization. Okay, you think that everything is going to be smooth, but the real is before you decide on your experiment or characterization, okay, you have to think about whether the instruments is available or not. Okay, so number one, the instruments is available or not, and whether the material that you decided to use is accessible or is not available or not. Okay, so for those of you who actually have decided based on what you already have in the lab, you already know that the instruments is working, then it's okay. But let's say in future you wish to do a project, okay, these are the things that you have to think about. One, of course, the money, whether you have the money to do to buy the materials. The other one is whether you have the money to pay for the services, okay? And the third one, probably the money to have contingencies. You need to send the samples to the other labs, for example, if let's say you cannot run the experiment by yourself, okay? Now, you might think that the instruments is available. Okay, I know, okay, the instrument is available. I know that it is there in UPM. I know that it is there in the faculty. You might need to check whether the instrument is working fine, one, okay. The other one is, if it is working fine at that moment, let's say at the time that you want to send your samples to the instruments, to the services, it broke down. Okay, what are the contingencies? Do you have contingencies? Do you have plan A, plan B, plan C? Okay, that is very important. That is based on my experience. I always have contingencies. If not, then I'll be dead meat. Okay, I have to answer the stakeholders. I receive grants, millions of grants. So if I do not answer to the stakeholders, then as I said, I'll be dead meat. They do not accept reasons like instrumentation breakdowns. No, I have to solve that issues from the beginning. I need to have contingencies. I need to know where to send the samples if the current instruments broke down. Okay, so that's why when you design first, let's say you design the experiments for the first objective. Let's say extraction of characterization of the natural products. Because I'm not in natural products, so I do not know. But you need to characterize. You send to GCMS, you send to NMR. Okay, and you think that you're going to complete that within a month. But by the time you want to send it, then you know that the instruments broke down. Okay, so now from 1 month, you have to wait until 3 months. So that's why when you design the first objective, and under that objective, you design the experimental design or the research design, you have to consider the time factor. Okay, because in the end, you will have to do what we call as Gantt chart. Okay, so now instead of 1 month, you design it to be a bit more than that, 2 months, because you are considering the factor of instruments broke down, or you need to queue, right? Okay, unless you are very rich. Okay, you have unlimited resources, you have a lot of money, so you can send to somewhere else. You know that there are NMR, GCMS available at somewhere else, and you just have to pay more. Okay, you just have to pay more, then it's okay. But if you think that you do not have enough resources, you have to queue, you have to follow the normal procedure, then you have to consider that in your research design. Okay, that will be sort of contingencies, time, and money related. Okay, and there are things about doing research in Malaysia. It's about the materials. Okay, so if you decide on to use whatever materials in the lab, okay, you need to justify, because in the end, let's say when during the viva session, viva is oral examination in the end, you'll be asked, for example, why do you use this material? And then you should not answer because it is the one that is available in the lab, because that's the wrong answer. Okay, you use it because you have justified in your research problem that material A, for example, is very good for your solution. Okay, but when you design your proposal and you forgot to check whether the material is already available, or you forgot to check if you order, how long will it take to arrive to Malaysia, then you'll be in big trouble. So checking or studying or asking information on the material, whether you can receive it as early as possible, how long will it take to arrive in Malaysia is very important. Okay, so that will be on instruments, on material. Okay, the other one also services. Okay, services means that sometimes the instruments no, it's not like instruments. Sometimes, let's say you're doing something related to drug delivery for cancer, for example, yeah? And you need to do animal study. So when you need to do animal study, you have to apply for ethic approval. So that ethic approval, because we are dealing with animal, that ethic approval will take some time. So you have to plan your research design. When are you going to apply for your ethic approval? let's say you do not need to apply because you just want to send it to the lab. For example, there are special labs who can do that services for you. So you have to check, okay, whether how much they requires you to pay, and then whether the services are accredited, because in thesis reports, you have to declare where did you do the analysis, what kind of material that you use, what kind of services, because when we write thesis, I'm sure that you have done it in your final year project before, during your undergraduates. In experimental section, you have to write the brand, the purity of the material, as well as the model of the instruments. So those are very important when considering experimental design. So how much, okay, how does it take? One of the most important is to anticipate, okay? Because all these are anticipating as well, yeah? Okay, contingency, okay, so the keywords just now, contingency, you need to have plan A, plan B, plan C while designing the experiment. Okay, let's say you choose method A, okay, and you check everything is there. You have all the required instruments, you have the oven, you have the all the things in the lab that you need is there, so it's fine. Okay, but what if it is not there? So it's a contingency plan, plan B. Okay, so as I put here, as a researcher like me, I have to answer to my stakeholders. Okay, I cannot just give reasons why I cannot achieve my objective. As a graduate student, means that soon you're going to you're going to do your small project. You have to answer to your supervisor, because you want to graduate, of course, in the stipulated time, yeah? The time given to you. So very important. That's why I said when you are designing this, you are designing your life as well, because your life will be there, will be in the experimental design. Yeah, a lot of time you are going to be in the research as well. So by the end of this unit, this discussion is you'll be able to plan suitable methodologies and procedures. Okay. Right. Okay, because based on that just now, you know your instruments, the availability of the instruments, availability of the materials and services, so you can decide on the suitable methodologies and procedures. Anticipate problems and challenges. This is very important. Okay, mitigate the anticipated problems. Once you anticipate, of course, you have the mitigation action as well. Okay, so how do we do this? How do we anticipate problems? So can we talk about this? What kind of problems that you will anticipate, that you can anticipate? Yes. No problem? Or you say, okay, I will deal with problem whenever it happens. That's what we usually do. Okay, we deal the problem whenever it happens, whenever it will, or whenever it appears. Okay, but for us, if we really want to finish it on time, we have to anticipate the problems. Okay, so it is related to your issues that you want to solve, so you know you should be able to anticipate. As for me, okay, because we have a different view in problems, we have different problems. I have a project that is not running. Why? Because the students keep quitting. They quit. After 3 months, they quit. After 3 months, after 1 year, they quit. So the project is not running smoothly. So these are the things that I have to anticipate, that the students quit the project. They move to the other projects, or they change universities. So how do I answer my stakeholders? I cannot just say that my students quit, so I cannot do the research, I cannot finish up the project. That is not acceptable, because the stakeholders will say, that is not my problem. Because when we proposed, we have to think about the mitigation issues, the mitigation plan. How do we mitigate those kind of technical problems? So my contingency is always to have more than one students to work on the same project. Okay? So that will be my mitigation or contingency. Okay. So every time I anticipate that this is going to happen, students quit, or they are offered a different, more interesting project. So in your view, what do you anticipate the problem will be? What do you foresee is going to be your problem? It's not like having something, oh, I'm very negative. No, this is a very good practice. Anticipating. Anyone? Related to your boss?
The delay of chemical.
Yeah, that one is very common. The delay in chemicals. Okay, arriving, especially in Malaysia, it took 3 months, sometimes 6 months to arrive. So if you think about this, you already knew it's going to happen. Then you have to, what we did among our researchers is we borrow first. Okay, okay, let's say my friend has that material, so I said I'm in the process of ordering, so may I use your materials for a bit, and then whenever that mine arrive, I just give it back to her or him. Okay, so that kind of contingencies that you can do. Okay, that if you have planned, but if you do not plan, then you just wait for 3 months the materials to come, then it's going to be a big problem. Okay, because you're working, so what do you foresee your future problems when you are going to complete your master dissertation?
Time, Prof.
Yes?
Time.
Time, yeah? Yeah, I can see that. That time is going to be your biggest problem, okay? Because we are human. Sometimes we are prone to sickness, having fever, fatigue, all this. So whenever that you design your experiment, you have to consider the human factor as well. Okay, you cannot think that you'll be working 100%. You can complete. So whenever that you're designing, okay, you know that you can do that. The instruments is there, material is there, and you'll be able to do that. Okay, if not, then some parts of it you have to outsource. And some parts of it you have to outsource. That depends on your discussion, your discussion with your supervisor. So that the relation between you and your supervisor at this stage is very crucial. When you design your experiment, it's not like you design it separately and then you just present it to your supervisor. No, it cannot be like that. Of course, from the beginning, which is the research problem, you have to engage your supervisor. And when it comes to the experimental design, you really have to sit down with your supervisor, because you want to have understanding, because your supervisor should know that you have a very limited time. You only can answer certain scopes, and if he or she requires you to do more, then you have to to be able to rebut or ask to tell her that, okay, this is beyond my scope, or if it is required, then we can do outsourcing. Okay, I hope that you understand when I say outsourcing. Okay, so these kind of things. Yeah. So experimental design is not just about chemistry. It's not just about the science, but it's a human factor as well as external factors like instruments, materials, accessibility to the materials, and because you are sometimes you outsource the services to other labs, you need to know whether the lab is accessible, the service is accessible to you, and you do not have to you need to have a long queue to wait. Okay, those things you have to talk to people, you have to ask questions. Okay, that's why when for me when we have a new students in the lab, among the first thing to do is to mingle with the former students so that you know how the systems work. So they must be considered in construct. Okay, as graduate students, those are the things. Yeah? So now let's talk about the science first, the fundamentals of how are you going to plan to do the method selection. Okay. Okay. So method selection, this is the one that you have done, okay, during your literature review. Okay. So if you decided to, again, I use nanomaterial preparation and characterization. So in designing the experiment, you should know that the techniques to confirm that it is nano structured has to be in your experimental design. Of course, the usage of HRTM, okay, to look at the nano size, FISM, to look at the nanostructure must be. If it is not be in the experimental design, then how are you going to prove that it is a nano? You have prepared a nanostructure material. That's an example. Let's say you are doing biofuel. Okay, you come up with a new way to produce biofuel. Okay, but in your experimental design, you should have a standard material, a standard biofuel to be compared with your prepared biofuel, because you are developing a new method. You need to validate with the standard method. So it should be in your experimental design that you should have a standard method which is pure, and you bought it, you buy it to be compared with. Okay, so you cannot in the viva session in the end, your examiner will ask, how confirm are you that you get a good biofuel? You do not have the answer because you do not compare with a standard material. Okay, so that should be in your experimental design, because you want to achieve your objective, right? The first objective, let's say, is to your general objective is to produce a biofuel from these resources, for example. So how do you know that you have achieved that objective? Is by comparing with a standard material. Okay, so there are, of course, we have various projects. So I can only give you examples that I know. Okay, when you design the experiment, design it by answering objective. Under objective one, this is what I will do. These are the experiments that I will do. And these experiments will confirm that I will achieve the first objective. Okay, objective number 2, you have another set of experimental design. I think, personally, if you do that, you will be safe. Instead of you just design experiment and you didn't bother to look at the objective. It should be objective oriented. Okay. If you have questions, just ask. Eh? Now, things to consider before designing experiments. course, method selection, whether the method is acceptable. Okay, you cannot just say, okay, I want to choose method A because it is available in the lab. It should be acceptable. It's a standard method acceptable to be used in that kind of experiment. Okay, you have to know. The other one is sampling. How do you plan to do the sampling? Okay, sampling does not mean, when I say sampling, it doesn't mean that you take samples, food samples or water samples. Those are samples as well. But if you have, again, let's say I'm using natural products, okay? Natural products, and you have extracts from the plants, and you are sending it to NMR or GCMS, for example. Okay, how to make sure that your samples are representative of what you have extract. Okay, so it cannot be just one extraction. Yeah? It has to be multiple extraction, and the sampling method is whatever samples that you have should be representative of what you're studying. Okay, it should be the same. Let's say you choose to have method 1 to produce it. Okay, so let's say you finish up the first sample. So you still use the method 1 technique to produce the second batch. Okay, so the second batch should have the similar characteristic with the first batch that you synthesize. Okay, so you need to think about the reproducibility of your sample, how reproduced your sample is. So can you imagine if we receive vaccine, the first batch is different quality, the second batch is different quality, then that will be a disastrous. We do not want that to happen. So that's why sampling is very important. How do we sample the product, the material that you produce? Or let's say you're doing samples from environment. So it's very important to understand how to sample. Okay. Again, if you have questions, just stop me. Okay. Sample processing. Okay. So whenever that you want to send for analysis, okay, is it suitable for NMR analysis? Is it suitable for Raman analysis? Is it suitable for FTIR analysis or any other analysis? So that is your responsibility, not the operator's responsibility. You have to check whether your sample is soluble, you use the correct solvent and all that. So it's your responsibility, and it should be in your research design. You understand your samples are going to be dissolved in what kind of solvent, because you have studied. You know that the sample is polar, so you have to use polar solvent. You know that you want to extract polar compound, so you have to use polar solvents. So those are the things that should be in your experimental design. It's you who are the one who knows about it. It's not the operators. It's not the one who run the machines. Yeah? Sample form. Yeah? So when we talk about sample processing, it relates to replicates and sample form, suitable forms to be run for analysis. So replicates means that you cannot just run a single experiment. It has to be replicates, minimum, I would say minimum of 3, but now sometimes minimum of 3 is not enough, because the data that you will obtain, we'll talk about data under data analysis. The data that you will obtain, you might have blunders. Okay, the results might be blunders. Okay, so if you have extra data, then you can have a good comparisons between the data. But if you have only a small number of data and you have a lot of blunders, then you'll be in big trouble. Okay. Sample form. Okay, sample form. Let's say you are doing something related to solids. Okay, solid samples, and you want to send it to FTIR, for example. So you must make sure that the sample is refined. You have to sieve the sample. Let's say you want to send it to particle size analyzer. You want to check the size. Of course, you have to grind it, sieve it to come up with a standard size of sample. Okay, those are the things that you have to consider. And these comes from your literature review. Okay. Interference. This is another big issue. Interference, because as I said, when I mentioned about biofuel just now, you tend to prepare biofuel from natural resources, from algae, for example. So it's not going to be pure. It's not going to be pure. So that's why you have to check, you have to validate, you have to compare your data with standard biofuel, a pure biofuel. So the interference means that the matrix, okay, the matrix means that if it is algae, you know that there are things in algae that you cannot avoid. So you have to think about this. Do I need to remove the interference? Do I need to process it before I run measurements? What will be the interference? What are the things that are going to interfere with the analysis, with the measurements? Okay, so this also should be in your experimental design, removing interference. How do I get pure compound? Okay, so those kind of things. Yeah? And now you have done, we are talking about the method that you choose. And then you have done a good job in sampling, sample processing. You have replicates, you have a good sample standard size or a very good dissolved soluble sample. And then you have thought about the interference, you have done purification as well, for example. And now it is time for you to do the measurement, right? Now it is time for you to do the measurement. So the measurement, there are things that you have to think about, which is the errors that might be contributed from the instrument itself. Because instrument works, but are they well maintained? Are the instrument in good condition? Can the instrument contribute errors? Yes. Okay. So that's why when you receive data, when you measure and then get the data, you really have to look at the data before you plot graph, you plot bar charts, or you are happy with your data. You have to look at the data, really look at it, and then do some statistical analysis, whether you have a good precision, whether you have a good accuracy, whether you need to do removal of outliers, or you have all blunders, you have to repeat again. Okay, so you have to look at this first before you do data evaluation. Sorry, you do reports, plotting data into a graph, and then from the graph you conclude the trend. Okay, so if you have a blunders in sets of data, when you plot the graph, of course, the conclusion will be wrong. Okay, the conclusion, the observation will be wrong. Okay, so this is the way that you should do for all the analysis. Okay, to go the steps to really understand and follow these steps, and then in the end, whether you think that you should repeat or you think that the data is acceptable and the observation, the conclusion is acceptable, then it's fine. That's why at the beginning of research characterization, one thing that I mentioned is about ethic. Ethic is not about ethic approval for do animal study, but ethic in doing research. Because I know things happens, everybody knows things happen, When you have a very limited time, you're very tired, and you look at the data, oh no, the data seems like it's not right. You know that it's not right. Then this is where your ethic plays a role. If you're very ethical, you will try to sit down and think about what's the next action. Okay? Don't just write strictly. Don't just directly use the data. Okay, right. The word reliability, reliability and validation. So I'm giving you an example. So everyone knows what because this is something happening right now. This is the usage of rapid test kit, COVID-19 rapid test kit. So how reliable the test is. Okay. So in Malaysia, well, we are thankful that we have actually quite a strict, stringent what we call evaluation steps before the kit can be accepted to be used in Malaysia. So it is evaluated by IMR, they validate with standard methods, and then after that, the medical device authority will approve the usage of that particular kit in Malaysia. But imagine if that is not happening. Imagine if that is not happening. So we just receive the kits, whatever country it is from, and then we use it. So it's going to have a disastrous effects. You have a lot of probably false positive, false negative, and you can imagine the chaos if that's happened. Okay, so that's why reliability and validation is very important in your experimental design steps. How are you going to check whether your data is reliable or not? You question yourself. How are you going to check? Okay, just now I mentioned about standard material. So probably standard material is something that you can use. Standard method. Okay, let's say you are developing new methods to remove metals, for example, heavy metals from water. So how do you know that you have obtained a very good removal? By checking with ICP, by checking with AS. Okay, so you know that yes, you have a very good removal ability for your material. And another way, let's say you have developed a new method to detect glucose. Okay, so how do you know that you have done a good job? Let's say you have a sample and you have done with your method. The same sample you send to a validated using a validated technique, so you compare the results. Let's say you want to sell the methods, so you have to send the sample, the same sample that you did just now, to accredited lab. Okay, you send the sample to accredited lab, and then you look at the results, whether you get a similar or you have to do the F test, the T test, check on the position as well as the accuracy. So it's a tough job. Okay, you are designing, as I said, you are designing your life. If you decided to do master dissertation in 1 year, so you are considering your life for 1 year. So you have to really look on the experimental design. Yeah, it's not just like I can't just copy from my seniors and paste it in my proposal. Yeah, it might work, but you should know better. It's going to affect your life. If it doesn't work, you'll be in big trouble in the future. So might as well you think about it first, anticipate the problems, because your time is not similar to your senior's time. It's not similar to someone else. You know better. All right. Okay. So you have questions? Okay, this one is the detail. Samples should be representative and compliance with standard techniques. Replications of analysis is a must. Okay. Ini just activities. Okay. Questions before we take a break? You are the one who's going to write. Yeah? This is a second assignment actually, but I want you to finish up the first. So I'm giving you 1 week to finish up your problem statement, objective, literature review, and objective. So this is the experimental design. Okay? Questions? No questions, okay? So if there is no questions, just take a 10 minutes break. Okay, 10 minutes means now it's 3:30. So 3:30, 3:40, can we start back? And I have Mr. Azizo. Okay. So when we receive data, okay, just now go back. When we receive data, we can do statistical analysis with the data, and we can do data analysis by using software. Okay, so Mr. Azizo will be talking about PCA, principle again. Apa Azizo? Principal component analysis.
Principal component analysis.
Principal component analysis. This is one way of how we can use the data to look at the trend. Okay, so Mr. Azizo will be giving an example of the projects that he has worked on to see how you can use principal analysis, PCA in short, to analyze your data. So we take a break, 3:40, and then we can have Mr. Azizo after that. All right, thank you. Yeah.
First is for data overview using PCA. It describes the correlation structure in X in which similarity or dissimilarity can be observed. Also, outliers can be detected. Second is for classification and discrimination using PLSTA or OPLSTA. We apply this when we need for further modeling of each group in order to fine-tune the analysis. It used the separate class models for classifying new observation. Third is regression modeling using PLS to determine the relationship between X and Y. From this analysis, we can predict how the X variable affect the response and how the X and Y variable correlate to each other. This table shows the difference between unsupervised and supervised analysis. Unsupervised analysis have no pure knowledge in which the algorithm is not provided with any pre-assignables or scores for the training data. It allows for unbiased view of data. PCA, ACA, and heat map are examples of unsupervised analysis. Unsupervised analysis learned from label training data helps you to predict outcomes for unforeseen data. For example, it can be used to study the relationship between NMR or MS or IR spectra and its bioactivity. The LSTA and PLS are examples of supervised analysis. Now we move on to PCA. PCA is a statistical procedure that uses an orthogonal transformation to convert a set of observation of possibly correlated variable into a set of value of linearly uncorrelated variable called principal components. PCA is one of the most important dimensionality reduction technique that is available out there. So what is dimensionality reduction? Let's assume you have a huge table of data. This data table is made up of multiple samples in row and variable in column. This variable is called X variable. X can be, for instance, the chemical shift of NMR. Because there are so many variables, it is not reasonably possible to understand the relationship between each different variable by drawing typical correlation plots. So we want to make this table into a smaller one while still keeping as much information as possible. So dimensionality reduction technique, what they do is they reduce the number of columns into 2 dimensions. For this to happen, there are many linear algebra involved. Let's say we need to take a picture of the UPM mouse. The question will be, where should we take the picture from? At which angle gives the best representation of the UPM mouse? The last picture is the best angle to take the picture because it gives the best information on how the UPM mouse should look like. This is what dimensionality reduction is. It's taking a picture of your data and trying to keep as much as interesting information from your original data in your picture. The information that you get from dimensionality reduction may not explain 100% of the original data. This is because some variables measured from our instrument are noise. In order to reveal what is the hidden information, the PCA model in matrix X is constructed by characterizing the most important variation of the data and eliminating the unstructured variation or the error. The fit of the model is measured by the percentage of explained variable. The bigger the percentage means the closer the model is to the original data. Otherwise, for poor explained variable, we can say that the data table is mostly consists of noise. First principal component of PC1. What you see here is X, Y, Z axis that represent variable. The red point are samples in the variable space. The whole data table constitutes a swarm of points in the variable space. Our goal in the PCA is to find the structure of this swarm. For this, the principal component axis D1 are rotated along the axis to find the best angle having maximum variation, and the objects are projected into D1. This gives the total variance, for instance, 53% explained by D1. Now D1 is rotated at a 90-degree angle to a new PC called D2 to explain the remaining variance. For example, for PC2, the explained variance is 17%. If you compare both PC, we see that the red object from PC1 are more spaced out the red object from PC2. This is why you see that the explained variance is larger than in PC2. PCA always try to find the maximum variance in the first component and find other PC to explain more variance. In this example, the first 2 PC explain 70% of the original data by combining PC1 and PC2. 53% plus 70%, you get 70%. The remaining of 30% does not contribute to the projection. How to interpret score plot? Score plot is a map of all samples. It shows grouping pattern and outliers. Distance between sample mean difference. Sample 1, 4, 7 are similar and they are different from sample 8, 9, 10 and 2, 3, 6. Close to the center means average sample. mean that sample 5 is a typical sample. Now we go to the interpreting loading plot. Loading plot is a map of all variables. It is useful to understand the correlation between the variable and interpret sample grouping. Start from center and point towards a variable. Variables close to the center are not well summarized. For example, sample D. Large loading, for example, sample C, indicate that variable has strong effect on the principal component. Variables along the same direction are correlated. For example, sample A and E. We can visualize loading plot in 3 types: loading scatter plot, loading column plot, and loading line plot. The loading plot I show you just now is loading scatter plot. I will show you later the example of loading line and column plot in case study. Interpreting score and loading plot. Side by side, these two plots very helpfully characterize all the observation in the data set. The two plots are complementary and superimposable, and a direction in one plot corresponds to the same direction in the other plot. This is an example of food data set. For this study, data were collected to examine similarities and difference of the food consumption between European countries and the possible explanation. The dataset consists of 16 samples, the European countries, and 20 variables, the common type food consumed in the Europe. This data matrix table shows you an example on how you can organize your data for any multivariate analysis. The sample is usually arranged in column and variable in row. Additional group information, metadata, describing the geographical location of each sample is adhered to facilitate data interpretation. Having metadata is indeed important for sample grouping. This class is only considered as the secondary observation and not Y variable, so it has no effect on the modeling. Okay, model evaluation. Principal component is referred here as A, and N is the number of samples used in this model. The number of PC that is taken into account to create the model was 3. The best fit or R2X cumulative indicate that 3 PCs explain a total of 64.8% of the variance. The remaining of 35.2% does not contribute to the projection. The first PC always explains the largest variance, and as you have more PC, this value decreases. Q2 is the estimate of the predictive ability on new data. A negative Q2 indicates your model is not all predictive. The R2 and Q2 value that you can expect is highly application dependent. Generally, if R2 is lower than 0.5, the data contains large amount of noise or irrelevant information in the data. In this model, the R2 value was 0.648. There are 16 points representing 16 countries. The score plot displays the first 2 principal components of a data set about the food consumption profile of European countries. These scores are called T1 and T2. Because we have metadata, here the countries are colored according to the geographic location of the capital city. The first 2 components explain 50.9% of the variation, 31.7 plus 19.2, you got 50.9%. Countries close to each other have similar food consumption profile, whereas those far from each other are dissimilar. For example, the Scandinavia are located together in upper right corner. These nations have similarity in food consumption. Belgium and Germany are close to the center of the plot, and this indicates they have average properties. There is no clear separation between Northern and Western Europe. The Mediterranean region are located together in the bottom left corner. The geographic locations seem to influence the food consumption pattern, especially between the Scandinavia and Mediterranean. From the loading plot, we would like to determine which variable or food item are responsible for the pattern seen among the countries. We would like to know which variables are influential and also how the variables are correlated. The loading vector in this plot are called P1 and P2. This figure displays the relationship between all 20 variables at the same time. Variables contributing similar information are grouped together. That is, they are correlated. The distance to the origin also conveys information. The further away from the plot origin a variable lies, the stronger the impact that variable has on the model. I have included the score plot here to help you to see which variable in the loading plot relates to the samples located in the same direction. The corresponding loading plot shows garlic and olive oil in one discriminating group of variables. These two provisions are often consumed in the Mediterranean countries. Crisp bread, frozen fish, and frozen vegetables are eaten extensively in the Scandinavian countries, while the Western European countries drink instant coffee and eat powder soup. Now, since the PCA model uses 3 components, we can investigate if the projection of PC1 versus PC3 will provide us with additional information on the food consumption pattern. We could see other patterns where England and Ireland can be distinguished from the rest European countries based on the consumption of tin soup, tea, and jam. This dimensionality of 16 times 20 data matrix was reduced to 3 PC to reveal the underlying grouping and pattern of food consumption among European countries. What is an outlier? Outlier is sample which deviates from the other samples in a model and may not belong to the same population as the other majority. Simca software draws the tolerance ellipse 95% confidence limit based on Hoteling's P2. Samples situated outside the ellipse are potential outlier, which may disturb the model. Diagnostic tool to detect outliers. The Hoteling T2 column plot displays the distance from the origin in the score space for each selected observation. It is common to have about 5% sample to lie outside the ellipse. We need to investigate the seriousness of this outlier. For this, we can use the Hoteling's T2 plot. Value larger than 95% confidence limit are suspect, and the value larger than 99% confidence limit are serious outlier. For example, this sample are larger than 99% confidence limit and is a serious outlier. In this example, are sample 3 and 4 outliers. The sample lies just outside the ellipse. The Hoteling's T2 value did not exceed 99%. Another diagnostic tool to determine the seriousness of this outlier are called DMOD X. DMOD X is an acronym of distance to the model in the X data and indicates how well an observation fits the PCA model. Sample with a DMOD X larger than discrete level, red dot lines, are outliers. If the DMOD X is twice the discrete value, they are strong outlier. In the DMOD X plot, sample 13 are different from their population with respect to the correlation structure of the variable. So, sample 13 is an outlier. Outlier to remove or not. When a sample has been detected as being an outlier, it is often interesting to try to investigate the reason. An outlier may be due to wrong labeling, measurement error, noise, or may indicate something scientifically interesting. Some outliers can be corrected or may need to remove. If in doubt as to whether to remove an outlier or not, compare the models before and after removal. The case study 1. This paper describes the differentiation of non-infected and G. boninens infected all palm roots at 14 and 30 days post-infection using GCMS-based metabolomics. This combination has provided a rapid approach in investigating the change in the metabolites variation of non-infected and infected all palm roots at 14 and 30 days post-infection. This table shows the metabolite identified in healthy and infected roots at 14 and 30 dpi. PCA was applied to understand the clustering features of the non-infected and infected samples and the metabolites contributing to the variability with the first two principal components explaining 79.2%. The score plot shows two clear separated clusters identified by PC1 without any notable outliers. In identifying the root extract metabolites responsible for each differentiation, PCA models were generated in loading plot. As we can see here, metabolites on the right side were found more abundant in infected sample, whereas metabolites on the left side were found higher in non-infected sample. Fatty acid and dimethyl ester could be acting as potent antimicrobial agent against pathogen. The highest concentration of the steroidal compound, this one, 14, 15, and 16, are related to the act as plant defense metabolite apart from performing as plant growth regulator and controlling the fluidity of plant membrane for alteration to change in temperature. The research showed that GCMS chromatogram of non-infected and G. boninens infected root exhibit different which were discriminated and clustered into groups to motivate data analysis of PCA. Case study 2. Now we go to the case study number 2. This work describes the differentiation of non-infected and G. boninens infected leaf using NMR-based metabolomics and PCA. The spectra shows the proton NMR spectra of all palm leaf, whereas this table shows the identified metabolite in healthy and infected sample at 14 days post-infection. Identification of metabolite variation of healthy and G. boninens infected all palm leaf was done by PCA. PCA was used to cluster the features of the samples and analyze the metabolites that contribute to the variation. Healthy and G. boninens infected all palm leaf could be discriminated clearly as shown in this figure. The first two principal components, PC1 and PC2, cumulatively accounted for 87.5% of the total variation. Separation of the healthy and G. boninens infected all palm leaf in score plot was achieved by combining PC1 and PC2. From the loading plot, the level of xylose, alpha-glucose, gallic acid, and acetylcysteine were more abundant in healthy leaf, whereas these metabolites were more abundant in infected sample. Sugars play an important role as carbon and energy source. Also, they have been recognized as signaling molecule of plant defense under biotic and abiotic stress. Accumulation of sucrose and fructose in G. boninens infected leaf should be related to their role as energy source for pathogen replication. In contrast, xylose and alpha-glucose were decreased in infected leaf, which indicate a part of the available carbon gathered in carbohydrate is consequently diverted to a secondary metabolism. The carbohydrate also biosynthesis. The high level of alanine in infected leaf was observed. It has been reported that alanine synthesis is increased to regulate cellular osmosis, which is decreased by high cellular carbohydrate levels under stress condition in infected plants. Meanwhile, accumulation of choline amount in infected leaf is associated to an increase in the synthesis of membrane components after pathogen infection. It has been reported that infected leaf induce collagen synthesis through the jasminic acid signal transduction pathway. Phenolics such as camphorol, 2,3,4,3-hydroxybenzoic acid, para-hydrobenzoic acid, and caffeic acid level were more prominent in G. boninus infected leaf. These metabolites were produced by plants to help in cell wall lignification, antimicrobial activity, modulation of plant hormone involved in defense signaling pathway, and scavenging of reactive oxygen species. Indole triacetic acid, number 9, was found higher in infected leaf. It acts as inhibitor of virulence gene expression in plant. It also functions as a chemical agent metabolite in plant defense against pathogen. Okay, this study described the metabolite profiling of chokgukjang, a fermented soybean paste during fermentation by GCMS and PCA. As shown in this figure, the chokgukjang sample taken at different fermentation time were clearly distinguished in the score plot generated by combining PC1 and PC2. The sample could be separated according to their fermentation period, mainly in the score of PC1. Referring to the column loading plot, okay, this is a column loading plot. The major metabolites contributing to the PC1 dimension were these metabolites. The level of this metabolite decreased as fermentation proceed. In contrast to those of 2-hydroxyglutaric acid, leucine, and tartaric acid, which did the reverse. The sample from 20 hours of fermentation could be more clearly discriminated in the score of PC2. This is PC2. This is a positive quadrant, negative, this one positive, and this one negative. Level of serine, methionine, lysine, phenylalanine, aspartic acid, and glycine increase as the level of most amino acid decrease in the early stage of fermentation and increase in the later stage. Level of fatty acid generally increase throughout the fermentation process. They conclude that PCA demonstrated that this metabolite were the major component that differentiate the sample according to the fermentation period. Okay, this journal explain about the discrimination of cabbage cultivar ground in different geographical areas using proton NMR-based metabolomics. Metabolomic analysis was performed to distinguish two cultivar of cabbage ground in different geographical areas, Korea and China, using proton NMR spectroscopy coupled with PCA. Okay, this is a loading line plot. To provide a comparative interpretation and visualization of the metabolic difference in cabbage according to the different growing areas, PCA was applied to the NMR spectrum dataset for each cultivar. The PCA score plot show fairly clear difference between China and Korea ground sample of both cultivar, indicating that cabbage metabolites are strongly influenced by the environmental condition of each cultivation area. From the loading plot, these metabolites were higher in Korean sample, whereas these metabolites were abundant in China samples. Okay, similar to this one. Okay, basic fact on PCA. PCA is an unsupervised classification. It reveals grouping, pattern, and detects outliers. It is an orthogonal projection of a data which reduces the dimensionality of a complex data set to find the underlying structure in large data set. This is done by linear transformation of a large set of variables into a smaller set of new uncorrelated variables called PC in such a way that the variance of the data in the low dimensional representation is maximized, thus preserving as much information from the large set as possible. It is a powerful visualization tool and exploratory tool capable of providing an overview of complex data and uncover the relationship between samples and variables and among the variables simultaneously. The first PC describes the most variance. As you go from PC1 to the final PC, the amount of variance explained decrease. PCA used two graphical tools. Score plot shows the map of samples, and loading plot shows the map of variable. In score plot, distance between samples means different. Those close to center or origin indicates they have the average properties. Those outside the 95% ellipse limit is a potential outlier. Sample far from its population may also be an outlier. Serious outliers may disturb model. Determine what needs to be done by investigating the Hoteling's T2 and DMOD X plot. The score and loading plot complement each other. The position of samples in a given direction in a score plot is influenced by variables lying in the same direction in the loading plot. Variables with large loading value far away from the center are most important in explaining the sample. I think that's all from me for today.
Thank you so much, Azizo, for helping me. Okay, so this is one of the tools that you can use to analyze data. All right, so of course, it's not for you to understand, to grab everything in one session, because for this kind of tools, Simca and there are other tools, is the best way to understand is for you to use the tools. So it's not for you to grab all the because Mr. Azizo has been using it for quite some time. But these are the tools that you can use to analyze your data. So you can see that, let's say you have a lot of data. So how do you analyze and come up with a conclusion? So thanks to the advancements now that we have a computer, this software that can analyze the data. So imagine that we have to look at the data with our own eyes and sort it out. So it's almost impossible. But with the help of software, you can sort the data, and from there you can have a very good conclusion, right? So, any question to Mr. Azizul?
Thank you, Mr. Azizul. I think we are going to learn this method in our statistic course.
Yeah, but similar because Mr. Azizul also is going to deliver the. Okay, so I may have a look because I have shared the slides. And if you have more questions, I think Mr. Azizul can at least answer another during your 5001, right? Azizul, when is your engagement with Dr. Jaafar?
Dr. Jaafar still not give me the date.
Okay, so you have the chance to listen to Mr. Azizul again and has a question. Okay, as I said, it's not something that you can grab in one session. You have to use the tools, then you can have more questions and all that. But it's a very useful tool. Yeah, any more questions? Questions on the research design as well? Yes, Firdaus, you.
How easy is to use the software? I mean, is it straightforward or need to do some? I mean, how easy is the software?
Not so difficult. Just follow the step, and you will get the result. Very simple software.
All right.
I believe Azizo means that you put in the data, right? And then you key the buttons.
Yeah.
Interpreting also requires fundamental understanding on how the software works. Okay? If you blindly put the data and you do not know what happened, it's not advisable, because at least you know some fundamentals, and then from there you develop. Okay, Firdaus? All right. So it's a very useful data. All right. Any more questions? Any more questions from you? All right. What about?
Software SIM card tu mahal tak?
Okay, Azul jawab Azul.
Software tu dalam dekat 20,000 kalau tak silap saya.
So, kalau kita nak pakai, kita kena pakai macam mana Cik Rizal?
Okay, software ni ada beberapa institut lah yang ada beli. Macam di dekat tempat saya pun ada beli juga software ni untuk kegunaan kegunaan lab lah. Jadi kalau ada siapa nak guna, mungkin ada collaboration lah untuk guna software ni.
Okay, baik. Thank you, Cik Rizal.
Alright, Azul, I think it's okay. No problem. You may have you can have another session with Mr. Azizul later. Memang mahal, it's expensive. But what we usually do, we share, okay, we share, and then Azul, like my student, it's not advisable. Let me stop the recording first.